Alpha-1 adrenergic receptor. Alpha-1 adrenergic receptors are G-protein-coupled receptors GPCRs associated with the GQ heterotrimeric G protein. And adrenergic receptors are subdivided into three highly homologous subtypes, i.e. AON A, AON B, and AON D adrenergic receptor subtypes. There is no AON C receptor. At one time, there was a subtype known as AON C, but it was found to be identical to the previously discovered AON receptor subtype. To avoid confusion, naming was continued with the letter D. Catecholamines like norepinephrine noradrenaline and epinephrine adrenaline signal through the AON adrenergic receptors in the central and peripheral nervous systems. The crystal structure of the AON B adrenergic receptor subtype has been determined in complex with the inverse agonist plus cyclazosin. Effects. The AON adrenergic receptor has several general functions in common with the ATWO adrenergic receptor, but also has specific effects of its own. AON receptors primarily mediate smooth muscle contraction, but have important functions elsewhere as well. The neurotransmitter norepinephrine has higher affinity for the AON receptor than does the hormone adrenaline. Smooth muscle. In smooth muscle cells of blood vessels, the principal effect of activation of these receptors is vasoconstriction. Blood vessels with AON adrenergic receptors are present in the skin, the sphincters of gastrointestinal system, kidney, renal artery, and brain. During the fight or flight response, vasoconstriction results in decreased blood flow to these organs. This accounts for the pale appearance of the skin of an individual when frightened. It also induces contraction of the urinary bladder, although this effect is minor compared to the relaxing effect of B2 adrenergic receptors. In other words, the overall effect of sympathetic stimuli on the bladder is relaxation, in order to inhibit micturition upon anticipation of a stressful event. Other effects on smooth muscle are contraction in uterus when pregnant. This is minor compared to the relaxing effects of the B2 receptor, agonists of which notably albuterol/salbutamol were formerly used to inhibit premature labor. Urethral sphincter, bronchioles, although minor to the relaxing effect of B2 receptor on bronchioles. Iris dilator muscle, seminal tract resulting in ejaculation. Neuronal. Activation of AON adrenergic receptors produces anorexia and partially mediates the efficacy of appetite suppressants like phenylpropanolamine and amphetamine in the treatment of obesity. Norepinephrine has been shown to decrease cellular excitability in all layers of the temporal cortex, including the primary auditory cortex. In particular, norepinephrine decreases glutamatergic excitatory postsynaptic potentials by the activation of AON adrenergic receptors. Norepinephrine also stimulates serotonin release by binding AON adrenergic receptors located on serotonergic neurons in the RAF. AON adrenergic receptor subtypes increase inhibition in the olfactory system, suggesting a synaptic mechanism for noradrenergic modulation of olfactory-driven behaviors. Other, both positive and negative inotropic effects on heart muscle. Secretion from salivary gland. Increased salivary potassium levels, glycogenolysis and gluconeogenesis in liver. Secretion from sweat glands. Contraction of the urinary bladder urothelium and lamina propria. Na plus reabsorption from kidney stimulate proximal tubule NHA3. Stimulate proximal tubule basolateral, NAK at pace. Activate mitogenic responses and regulate growth and proliferation of many cells. Involved in the detection of mechanical feedback on the hypoglossal motor neurons, which allow a long-term facilitation in respiration in response to repeated apneas. Signaling cascade. AON adrenergic receptors are members of the G protein-coupled receptor superfamily. Upon activation, a heterotrimeric G protein, GQ, activates phospholipase CPLC, which causes phosphatidylinositol to be transformed into inositol trisphosphate IP3 and diacylglycerol DAG. While DAG stays near the membrane, IP3 diffuses into the cytosol and to find the IP3 receptor on the endoplasmic reticulum, triggering calcium release from the stores. This triggers further effects, primarily through the activation of an enzyme protein kinase C. This enzyme, as a kinase, functions by phosphorylation of other enzymes causing their activation, or by phosphorylation of certain channels leading to the increase or decrease of electrolyte transfer in or out of the cell. Activity during exercise. During exercise, adrenergic receptors in active muscles are attenuated in an exercise intensity-dependent manner, allowing the B2 adrenergic receptors, which mediate vasodilation, to dominate. In contrast to adrenergic receptors, adrenergic receptors in the arterial vasculature of skeletal muscle are more resistant to inhibition, and attenuation of adrenergic receptor-mediated vasoconstriction only occurs during heavy exercise. Note that only active muscle adrenergic receptors will be blocked. Resting muscle will not have its adrenergic receptors blocked, and hence the overall effect will be adrenergic-mediated vasoconstriction. Ligands. Various heterocyclic antidepressants and antipsychotics are adrenergic receptor antagonists as well. This action is generally undesirable in such agents and mediates side effects like orthostatic hypotension and headaches due to excessive vasodilation.